biological system is not one thing. It is chemistry. It is proteins. It is gene expression. It is cell state. It is binding affinity, toxicity, sequence, structure, dose response, and context. Most of the time, when we talk about artificial intelligence in biology, we compress all of that into one vague phrase: drug discovery. That phrase hides the real problem. Drug discovery is not a single task. It is a chain of tasks. A molecule may look plausible in a model and fail in a dish. It may bind in a dish and fail in a cell. It may work in a preclinical system and fail in humans. It may be biologically active and still be impossible to manufacture, unsafe to dose, or too fragile to become a medicine. So the useful question is not whether a model can produce a promising biological answer. The useful question is what kind of biological relationships the model can represent, and where the validation system takes over. That is why MAMAL is interesting. MAMAL stands for Molecular Aligned Multimodal Architecture in Language. The paper describes a biomedical foundation model architecture built to represent several biological modalities in one structured system: small molecules, proteins, antibodies, gene expression, cell state, protein interactions, and scalar biological measurements. This is not just a larger language model pointed at biology. The architecture matters because biology does not arrive as one clean text stream. A small molecule may be represented as a chemical string. A protein may be represented as an amino acid sequence. A cell state may arrive as an expression profile. A binding measurement may be a scalar value. A drug response task may require the model to relate a cell line and a molecule, not merely summarize either one. MAMAL's premise is that those entities should not live in isolated modeling silos. They should be represented in a shared prompt and token system so the model can learn relationships across biological scales. That is the strongest version of the story. Not artificial intelligence cures disease. Not a model replaces the laboratory. The stronger claim is narrower and more useful. Biological artificial intelligence becomes more useful when it can connect the modalities that biology itself connects. The paper reports performance across 11 downstream benchmarks. The hardened result is important. It is not an 11 out of 11 sweep. Under the paper's own rule for what counts as state-of-the-art, MAMAL reaches state-of-the-art performance on 9 of 11 tasks, with 2 results better described as comparable or slight. That correction matters because it keeps the episode honest. The useful story does not need inflated numbers. The benchmark set spans several task types. Some are small, molecule classification tasks. Some use gene expression and small molecules together for cancer drug response prediction. Some involve antibody or protein sequence tasks. One drug-target interaction task combines protein and small molecule information. The pattern is the point. The model is being evaluated where biological meaning depends on relationships between different kinds of objects. A cancer drug response task is not just a molecule task. It asks how a molecular intervention relates to a cellular state. A drug-target interaction task is not just a protein task or a chemistry task. It asks whether those two entities meet in a biologically meaningful way. An antibody infilling task is not a clinical antibody program. It is a computational test of whether the model can recover or generate biologically structured sequence regions. That is the opportunity and the trap. The opportunity is that a model like this may make biological prediction less fragmented. Instead of building a different narrow model for every modality, the system can represent multiple entities and scalar values in one structured biological language. The trap is that benchmark success can start to sound like biological proof. It is not. The MAMAL paper demonstrates computational benchmark performance. It does not demonstrate a new wet lab result. It does not show a therapy working in animals. It does not show clinical benefit. It does not prove personalized medicine deployment. It does not mean that the model's connections are biologically real until experiments test them. That boundary is not a minor caveat. It is the central lesson. The model can connect biological modalities. Biology still decides whether the connection is real. This is also where the CHAI discovery evidence becomes useful, but only in a bounded way. The CHAI source hardening pass did not support the stronger claim that CHAI-3 has publicly demonstrated a full closed-loop system where molecular generation, experimental validation, and model learning recursively improve each other. The public Chi-3 evidence was mostly commercial and partnership language. The stronger experimentally detailed source was Chi-2. But Chi-2 is still useful evidence for this episode. Its reported antibody design work shows computational generation followed by small batch wet lab validation. Instead of screening enormous libraries, the system selected a small number of generated designs per target and then tested them experimentally. The important lesson is not that drugs are now produced in hours. The important lesson is that when generation becomes faster and cheaper, the bottleneck moves downstream. Binding still has to be measured. Specificity still has to be checked. Developability still matters. Manufacturing still matters. Preclinical and clinical evidence still matter. Regulatory proof still matters. In other words, faster generation does not eliminate validation. It increases the value of validation. MAM Mammal and Chi therefore reinforce the same QCS discipline from different angles. Mammal shows why cross-modal biological representation may matter. Chi shows why computational candidate generation can make experimental validation more central. Neither one makes the laboratory disappear. That is a better story than the usual artificial intelligence drug discovery headline. The headline says, "The model found the answer." The system story says, "The model changed which questions can be asked quickly, and the validation system now has to decide which answers survive contact with biology." This distinction becomes especially important in biology because biological systems are not static engineering drawings. They are context-dependent, adaptive, and messy. A relationship that appears in a dataset may fail under a different assay condition. A protein interaction may be measurable and still irrelevant to disease. A molecule may have the desired effect and unacceptable off-target behavior. A response in one cellular context may not generalize. This is why multimodality is valuable but dangerous to oversell. A model that can jointly represent chemistry, proteins, gene expression, antibodies, and scalar measurements may help scientists reason across more of the biological stack. It may help prioritize candidates reason across more of the biological stack. It may help identify relationships that single modality tools miss. It may compress parts of the search space. But the more powerful the representation becomes, the more important it is to preserve the boundary between representation and reality. A benchmark is not a patient. A predicted interaction is not a therapeutic effect. A binding classifier is not a medicine. A generated antibody sequence is not a drug candidate ready for clinical use. This is not skepticism for its own sake. It is what makes the technology usable. If we pretend the model replaces validation, the story becomes hype. If we understand the model as a way to connect biological modalities and focus validation, the story becomes infrastructure. That is the QCS frame. Biology is becoming more computable, but it is not becoming less biological. The near-term change is not that artificial intelligence skips the hard parts of drug development. The change is that models may move more ideas to the point where the hard parts arrive faster. They may create more hypotheses, more candidate molecules, more cross-modal predictions, and more reasons to expand the experimental systems that can evaluate them. That shifts the bottleneck. The bottleneck is no longer only search. It is also validation capacity, wet lab assays, target biology, manufacturing, animal studies, clinical trials, safety evidence, and the organizational ability to decide which generated possibilities deserve scarce experimental attention. Mammal's contribution is to make that shift visible at the representation layer. It asks what happens when biology is modeled less as isolated datasets and more as connected entities inside a shared system. The answer is promising, but bounded. The model can connect the modalities. Biology still decides whether the connection is real. For more evidence, first biomedical analysis, subscribe to Quantum Convergence. Quantum Convergence Bio.